Hi everyone, and welcome to our video on diabetes. This is part of our Module 5 Section 4 series looking at hormonal control. So what we're going to be looking at in this video is 5.1.4e, the differences between type 1 and type 2 diabetes mellitus, and 5.1.4f, the potential treatments for diabetes mellitus. First thing, let's understand what this diabetes mellitus thing actually is. And quite simply, it's a condition where the body is no longer able to control the blood glucose concentration. So because of this lack of control that we have, then anyone suffering from diabetes mellitus is going to be at greater risk of things like hyperglycemia once they've eaten foods, particularly those that are rich in sugars and carbohydrates. Or on the flip side, they're at greater risk of hypoglycemia after they've carried out exercise or they're carrying out a period of fasting. Now, there are two types of diabetes that we need to know about. Type 1 diabetes is also known as the insulin-dependent diabetes. Sometimes we call this the juvenile onset diabetes because it normally begins in childhood. And there are two kind of key causes of type 1 diabetes. First one is an autoimmune response which has then destroyed the beta cells within the pancreas. And of course our beta cells are the ones responsible for releasing insulin. So if our beta cells have been destroyed, we've got no cells to release that insulin. Second potential cause may be from certain viral infections. So autoimmune response or viral infection, which is basically going to lead to our body's inability to produce the insulin we need. Now, because those beta cells have been destroyed, then quite clearly we can't synthesize the insulin, and therefore we're not going to be able to store excess glucose within our bloodstream as glycogen. So what that means is that excess glucose is going to remain circulating within our bloodstream and give us high blood glucose concentration for an extended period of time. Now, that's obviously not great news for us because if we've got high blood glucose concentration, that's going to be affecting a range of other organs in the body and causing harm. When, however, the blood glucose levels fall, we have another problem because we don't have any glycogen, so we've got none of that storage molecule present, and therefore we've got nothing that we can release glucose from. Therefore, we end up with hypoglycemia and the issues that one can lead to. In terms of how we're going to treat type 1 diabetes then, this is typically done via an injection of insulin. Obviously these days we do have some rather fancy new or technological alternatives to someone sitting there with a needle and measuring out themselves. We do have things like the insulin pumps, which are obviously used by a number of students these days, and that actually connects onto mobile phones and everything, very clever these days. Now, obviously in order to work out the correct dose for your insulin injection, we have to know how your blood glucose is faring. So you have to do that little finger prick test and work out the blood glucose concentration first of all, and then you know how much insulin to administer. It's not just a case of you would just give yourself exactly the same dose of insulin every single time, otherwise that could end very badly. Other alternatives to the injection other than the pump, we could have islet cell transplants. So remember, the islet of Langerhans is where we've actually got those beta cells. So if we did a transplant of those cells, we can obviously replace them. Or we could do a whole pancreas transplant. If we replace the pancreas, we've replaced all of those lost cells. One area of research that could be very promising in the future is using stem cells. Now, the idea of using stem cells here is to actually grow new islets of Langerhans, and that's then going to obviously replace those beta cells that were destroyed by the autoimmune response or the virus. Now, those stem cells are typically going to come from the bone marrow or placenta, and what we're then able to do is obviously cause them to undergo their differentiation into those islets of Langerhans cells, the beta cells that we really need. What we do know is that from this research in mice, we've actually discovered these little precursor cells. And those precursor cells can be developed into a variety of stem cells. So what they're currently looking at is, do those same precursor cells exist in the human pancreas? If so, could we use them to produce those new beta cells? Because if the answer to that is yes, then obviously what we can do is use these stem cells to then reform those beta cells that have been lost and hopefully cure diabetes, which would be fabulous. The other type of diabetes is type 2 diabetes. This is sometimes known as non-insulin dependent diabetes and is often associated with a later onset in life. So the causes for type 2 are where there's just insufficient quantities of insulin being produced, or your body's not responding to insulin the way that it used to. So there's two kind of main causes there. What we tend to find is that those people with type 2 diabetes have this permanently higher blood glucose concentration. And this is where that risk comes into their organs and also circulatory system, because what we're going to see is a range of different areas getting damaged by that high blood glucose concentration. There are certain risk factors that make you more likely to develop type 2 diabetes later in life. Obesity is one of the main ones, and as we know, there's ever increasing numbers of people classified as obese, particularly in our developed countries like the UK, etc. And therefore the likelihood is we're going to see much greater numbers of type 2 diabetes cases in the future. We also have quite a sedentary existence these days. Very few people are actually taking regular exercise. The sofa and games consoles, mobile phones, etc. do have a strong pull versus a treadmill. In terms of our diet, it's a lot of sugar. If you are one of those teenagers that drinks those absolutely atrocious energy drinks, please stop first of all and just read the label. See how much sugar they're packing into those things. That is not good for you folks. And the sad news is that there's a lot of other processed foods that we are consuming with a lot of sugar in there, and that's not good for the risk factor for type 2 diabetes later in life. There's also family history. If you do have people in your family that have type 2 diabetes, then you are potentially at greater risk of developing it yourself. And then there are certain ethnic links, with Asian and Afro-Caribbean folks tending to be a little bit more prone than others. In terms of how we treat type 2 diabetes, it's typically by lifestyle changes. So we know obesity is a risk factor, so lose some weight. We know that a lack of exercise is a risk factor, so exercise more. And we know that this high sugar diet is a risk factor, so change your diet. There's a range of things that we can do that are actually really simple and therefore are going to dramatically reduce your risk of developing type 2 diabetes later in life. So take note, harness them now, avoid type 2 diabetes later in life. If of course lifestyle changes are not going to be sufficient, there can be medications which can be given to people which reduces the amount of glucose released by the liver or boosts insulin release, depending on obviously what your issue may be exactly. If we're talking about severe cases of type 2 diabetes, you may well require injections of insulin. So obviously it's not just a case of we can modify it all through lifestyle changes and occasional medication. Sometimes the need for insulin injections is going to come into type 2 as well. But predominantly type 1 is the insulin injections, type 2 lifestyle changes. Now, in terms of where this insulin that we're injecting into all these diabetics comes from, in the past it was a delightful source. It used to come from the pancreas of pigs that were slaughtered in the abattoirs. So when the pigs were taken for all the bacon, etc., then they would take the pancreases and then basically grind them up and extract the insulin. Lovely. Now, quite clearly, there's going to be a few people who have issues with that in life. And what we've looked at is coming up with a better idea, and this is genetically modified bacteria. And in theory at GCSE, you had a look at genetically modified E. coli that are producing human insulin. And the big advantages of using this genetically modified bacteria for insulin production as opposed to bits from pigs' pancreases. Firstly, it's producing human insulin because we're taking the gene for human insulin from a human and we're inserting it into the bacterial genome. Now, because it's human insulin we're making, it's going to be faster acting and more effective than supplementing a human with a different species insulin. We also have less risk of infections because we're not having any animals involved in this process. There's less risk of developing tolerance to something that is, to all intents and purposes, a foreign substance being injected into your body, and less chance of rejection for those same reasons. It's also cheaper. Bacteria don't really need much to live, and therefore it's far easier and cheaper to get this insulin from the bacteria than it is from larger animal species. Because we're talking bacteria which replicate very quickly, then that means that if we've got a sudden increase in demand, we can increase the amount of insulin produced very quickly from those GM bacteria because they replicate roughly every 20 minutes. So we can ramp up that production very quickly. And of course, because we are talking about bacteria, no one really stands on a picket line waving a placard about the rights of an E. coli cell, because no one particularly cares about E. coli, let's be honest. People will have greater issues if we are using animals that are quite cute at times. Therefore, we've got fewer ethical and moral objections. Obviously, you can also go down the route of religious objections, because certain religions obviously will have big issues with taking substances from pigs. And therefore, by using this GM bacteria, using obviously human insulin there, avoids all of those problems. As always, I hope you found this video useful, and don't forget to subscribe to the channel so you can see when the next video is uploaded. And of course, head on over to the website where you can find a range of other resources to help you in your A-level biology studies.